I'll be speaking today about some of the work that was started by the time I got here at South Carolina, but I've since taken it on, and we've seen some of our collaborators leave, like Brielle Stark. So I want to take a minute to acknowledge her work in this, and as well as Dr. Stephen Wilson, who is at Vanderbilt, who also had a part in this project. And I'm going to be talking today about neural synchrony during naturalistic perception in listeners with aphasia, and how neural synchrony relates to comprehension ability in our participants. So, as we're all very well aware, aphasia can be associated with deficits in language, be it in speech production or in comprehension. And considering what we know about the ventral stream and speech processing, that this is a very sparsely organized across the brain, we know that depending on the degree of damage or to specific regions, we can see a different etiology or origin in comprehension deficits across aphasia types. And prior to what I'll be talking about today, we focused primarily on investigating single modalities of comprehension ability. So utilizing very tightly controlled stimuli like the sentence that you see here, and utilizing controlled tasks and running basically a subtraction analysis to isolate the function that we're interested in, right? And trying to localize certain areas of the brain and relating that to a specific function. But recently, and this has been really helpful in letting us understand these neuroanatomical models and create neuroanatomical models of comprehension and language at large. But there's been recent postulations that this is not necessarily generalizable to naturalistic comprehension ability, and that we're not looking at the bigger picture during these kind of experiments. So that brought me to kind of the iceberg question that I call it for this project, in that we have a really good idea of the surface level of what's going on at each individual subtask of comprehension, but we don't really know how all of these come together into a broader comprehension mechanism, particularly during discourse and naturalistic comprehension tasks. So just to get us started, I want us all to be on the same page of what I mean by naturalistic comprehension, because when we talk about more typical neuroimaging studies, higher level comprehension usually stops at the sentence level, and it's really tricky to study more lengthy dynamic comprehension tasks like the ones that we're going to be talking about today. So generally, when we think about higher level comprehension in sentences, for example, we see that this is really organized across a broad range of brain areas, including regions within the temporal lobe, but also to some degree, some domain general regions in the frontal lobe as well. And people have suggested that this could be due to the recruitment of some higher level cognition ability, like theory of mind and working memory, for example. But when we increase, the length of and complexity of some of these comprehension measures and look at more discourse activity, for example, we see that we have even broader activity within across all of these regions. And though the necessity of these regions is not really clear, and there's been great debate on this in one really great review by Fedorenko in 2014, this is still nonetheless true that we do see some kind of activity of these regions during higher level tasks like naturalistic comprehension. So in looking at that kind of task of naturalistic comprehension, we move away from these more basic block level designs of specific stimuli and utilize more dynamic naturalistic designs, what they're called, where they have a greater length and no overt task that the participant has to do, rather than giving them a button press and having them indicate whether a sentence is grammatically correct, for example, or identifying a picture that relates to the sentence they listen to. They're actually just asked to sit back, listen, and attend to the video that they are presented with. And those videos can be short film clips that last like 10 to 15 minutes in length, or it could also be a full-length movie that's an hour and a half long, depending on the task. And there have been people that have utilized many of these different paradigms. The one that I'll be talking about today is short TV clips. So these are going to be 12-minute length or 10-minute length videos, and we looked at 2 of these. So I'll explain that a little bit more as we get into the specific design of the study. But with a different stimulus that we're utilizing, we also need to utilize a different analysis technique. And some folks have utilized what's called an intersubject correlation approach, or ISC analysis. And that basically makes the assumption that the brain activity across time can boil down into 3 main signals: what's called a consistent signal, or what's being specifically elicited by the stimulus that they are presented with. And that's thought to be consistent across individuals that attend to the same movie. So in this figure, we have 2 individuals that are watching or listening to the same movie. And the consistent response is referring to the bold signal that's going to be generated just basically from them attending to the stimulus. Of course, we have some degree of idiosyncratic responses based on how they individually are going to emotionally respond to the stimulus, for example, which is going to be different across individuals. And then we also have spontaneous or erroneous response that goes into, just general error terms of somebody drifting off and thinking about their grocery list, for example. But the main point here is that with intersubject correlation analysis, we're trying to measure the degree of similarity between individuals. And to do that, I pose this comparison of it actually being very similar to functional connectivity approaches, where instead of looking at within a single individual at multiple regions and seeing how well 2 areas talk to each other, we're looking at the same region or voxel within 2 or more individuals that are attending to the same stimulus. So we'll essentially output the time series of bold activity within each voxel or region and compare the same regions across individuals. So it ends up looking like a correlation, actually. So this ends up being a Pearson correlation where it's between -1 and 1, where 1 is more similarity between individuals. And 0.6 is pretty standard within areas that show relatively consistent synchrony. And we'll talk about those areas here in a second. So again, this correlation measure is to get an idea of how similarly in the brain more than one person is responding to the same task or the same stimulus. So there have been groups that have looked at this, particularly starting within the vision literature by Hassan and colleagues in 2004, but since has been utilized in language literature. But we've seen that these studies actually only utilize healthy control individuals and trying to relate their ability to comprehend a task or a stimulus and relating this to neural synchrony. So there is some question of whether or not this is actually a mechanism that is important for comprehension, or it's just a function of them attending to the same auditory-visual stimulus. So that brings me to the aims of the present study. So I wanted to take a closer look at this quote unquote mechanism and see if we can actually relate this to a clinical population. Of course, we're going to run into some issues given the fact that we have a lesion to some different areas within different individuals. And I'll talk about how I handled that within this data. But I first wanted to look at how a lesion can impact the intersubject correlation or overall synchrony in non-lesioned regions. So thinking about intact left hemisphere areas and of course, spared regions within the right hemisphere, as we know that comprehension is a bilateral task. And then I wanted to look at how or if intersubject correlations were associated with comprehension ability as per some offline comprehension tasks. So not specifically about the stimulus that they're watching, but offline measures that can be translatable to clinical application. So within our inclusion criteria, I do want to take a second and note that given COVID, we had to take a pause on data collection. So I'm only able to report on 12 neurotypical controls and 16 individuals with aphasia. So just keeping that in mind. But all of the participants followed similar inclusion criteria in that they had to be within normal limits for hearing thresholds with no correction because they were going to be in the scanner listening and attending to a task. And then our group with aphasia all were within the chronic stage of recovery, had aphasia as determined by the WOB. And we wanted to ensure that they had some degree of in-tact comprehension ability. So one of our inclusion criteria for the aphasic group was that they scored within, or at least a 4 out of 10 on the WOB comprehension subtest. And I can speak a little bit to that a little bit later if anyone has questions. So for study procedure, we collected T1 and T2 images. And for the naturalistic paradigm, it's very similar to a resting state scan, but instead of asking the participant to close their eyes and relax, we instructed them to keep their eyes open, of course, and attend to the videos that they saw. And the videos that they were presented with were 2 short abbreviated clips that were about 10 minutes in length of an old TV show, Freaks and Geeks. I don't know if anybody's familiar with that. And then we also made sure that they could hear above and beyond the scanner noise. So we gave them volume checks before playing the video clips. And then behavioral assessments that we were interested in, we gave them, of course, the WOB, as that was an inclusion criteria. And I was interested in relating global intersubject correlation scores with WOB severity scores. But also wanted to look at discourse comprehension or higher level comprehension tasks and see if subscores from the DCT were associated with intersubject correlation values. So those were 2 of our main outcome measures. So to look at this, I utilized the ISC toolbox by Calpe and colleagues in 2014, which is a freely available software, which is really, really helpful. But of course, we had to adapt this a little bit given the nature of the lesion, as this toolbox was developed for neurotypical individuals. But this toolbox allows for 2 main approaches for intersubject correlation analyses. And I want to take a second to explain that pretty clearly so that you guys understand what we did here. But the first approach I utilized was a pairwise intersubject correlation approach. And this was used to identify primarily seed regions within the controls that show relatively strong synchrony across neurotypical individuals, so that we could look at those specific seed regions within individuals with aphasia. This also allowed for us to run group comparisons, which is similar to, which is often done in studies that utilize intersubject correlation. So that was merely just an addition. But to run a pairwise intersubject correlation analysis, it looks a little something like this. So we had our control individuals and our individuals with aphasia, and we just pair up each individual within each group and then have an average map that showed average intersubject correlation across the group. And again, from this map, we identified seed regions that showed peak. So within each ROI that was identified as having strong synchrony across controls, I selected the peak voxel within each region and then dilated that by 5 millimeters to get a seed area for us to look at for regional synchrony for the aphasic group. And then similarly with the aphasic group, we paired up each individual. Notice that we have a lesion, of course, for every individual, which is going to be different. Within those lesioned areas, I assigned them NA values, so they weren't included in any intersubject correlation analysis. And then essentially you get an average mask, which of course I'm not interested in the left hemisphere too much, as this is to be expected that we don't have great synchrony within a clinical population who has a variety of different lesion profiles in the left hemisphere. But it is interesting to see that in the right hemisphere, which is intact, that they do have overall less synchrony. The second approach that we utilized was a leave one out intersubject correlation approach, which is very similar to previous studies that have used intersubject correlation analyses, in that this is used to look at how an individual is synchronizing to a group average. So this gives us a global intersubject correlation value, so looking at the average synchrony score for each individual. And then we also are able to calculate regional intersubject correlation values within each seed region that we identified from the previous analysis above. And that looks a little something like this. So for the control group, each individual each individual control was synchronized or correlated with the average group of the remaining control individuals. And then they each get a mask that we can calculate the global average for each individual and calculate the regional intersubject correlation within each individual scan. For the individuals with aphasia, as this was the piece that we were most interested in, as we can relate individual variability in their behavior to global intersubject correlation and regional intersubject correlations. We calculated the average bold activity for all control individuals and then measured the intersubject correlation between each individual with aphasia and the control group average. And you can see some examples that we have down below, where in red we have the lesion in areas that are shown in color, red being greater synchrony in certain areas and blue meaning less synchrony in certain areas. So that brings me to the results. We have, here's just a quick overview of some of the demographic information for both groups. Again, only 16 individuals with aphasia and 12 controls. We had about a 70 on the Western Aphasia Battery out of 100. So overall aphasia severity was pretty mild. And this is likely due to the fact that this was a auditory verbal comprehension was a criteria for inclusion there, in that they had to score above a 4 on this. And then our variability on overall comprehension is not too bad, but it would be beneficial to have a greater variability there. Here's a quick look at the lesion overlay map, where the greatest degree of overlap was within the arcuate fasciculus. Okay, so for our first analysis, again, this was to examine group differences and look at certain regional differences between groups. Within the intersubject correlation toolbox, I utilized a voxel-wise intersubject correlation analysis. So looking at each voxel rather than in regions of interest, and used multiple comparison correction with 5,000 permutations, where voxels that were less than 0.01 for their p-value were considered significant. Okay, so for the neurotypical map, so just to orient us, the cooler colors indicate a less overall synchrony, and warmer colors indicate a positive synchrony of 1. And this is a range from -1 to 1, of course. And we saw that there was relatively high synchrony for the neurotypical group in bilateral superior temporal gyri, bilateral medial occipital sulcuses, and then primary visual cortex in the left hemisphere. Here's a look at our group with aphasia, where we have synchrony within the right superior temporal gyrus, primary visual cortex in the right hemisphere, and bilateral medial occipital sulcus. To look at the group differences, we didn't have any areas of synchrony that were greater within the aphasic group compared to the neurotypical group, but we did have, of course, greater synchrony within the neurotypical group in left hemisphere superior temporal gyrus, and some other intact regions that were not impacted, that were not included in the lesion for the aphasic group, but did show overall greater synchrony within the neurotypical group compared to the aphasic group. And that was within the left superior temporal gyrus and bilateral medial occipital gyri. So from that analysis, again, we identified seed regions, and to get a measure of how well each individual with aphasia is performing to the control group, we compared each individual with aphasia to the control group average, and then each individual with control to the control group average, using the leave one out approach in order to get an idea of a degree of difference between the two groups. This also allowed us to calculate global intersubject correlations within the controls and regional intersubject correlations for within the controls as well, or from the controls for each individual with aphasia. And here's a look at those. So again, these are going to be the leave one out analysis measure, where this is a very similar map to the one we just looked at. But what's different here is that this is each individual with aphasia compared to the control group average to see how similarly each individual with aphasia is performing to the control group average scan. There was a statistical difference between the two groups in that the group with aphasia was not as synchronized to the control group as neurotypicals were to their own group. And then from that, we had 10 seed regions that we identified from the prior analysis and allowed us to look at individual regions and synchrony within these regions and how it relates to behavior. And those regions are listed here. Bilateral, these are areas that were synchronized within the neurotypical group. So the left hemisphere and right hemisphere calcarine sulcus. We had left inferior frontal gyrus, right parietal, interparietal sulcus, left precentral gyrus, right hemisphere angular gyrus, right hemisphere cerebellum, superior frontal gyrus, and then bilateral superior temporal gyrus. So first, I was interested in looking at how global or average intersubject correlation within each individual with aphasia was associated with behavioral scores. So the behavioral scores that we were interested in were discourse comprehension subtests, which included main ideas and details, as well as an overall measure of aphasia severity. So I ran a multitude of correlations controlled for multiple comparisons, but we didn't see any relation between overall average intersubject correlations and any behavioral measure of interest, let alone lesion volume or months post onset, for example, as well, which initially was not the most exciting finding, but looking within the literature that has utilized global measures of intersubject correlation and tried to relate this to behavior, it's been suggested that this function of, or this mechanism behind intersubject correlations is not necessarily a global relationship, but is actually more region specific. So that brings me to the regional analysis. Here's a look at each individual with aphasia, and within each ROI, we calculated the average intersubject correlation for each seed area, and you can see those here in different colors, where a -1 is going to be our unsynchronized areas. +1 is very synchronized between each individual with aphasia and the control group average. Okay. From this, of course, we had 4 areas within the left hemisphere that were identified from our control group. Within each individual with aphasia, of course, we have participants who have a different degree of damage to each area. So to control for that, we calculated the intersubject correlation within each seed region and regressed out the proportion damage to each ROI. And from this, we also had a few participants who had a great degree of damage to the seed regions. The inferior frontal gyrus and the superior temporal gyrus within the left hemisphere both had 5 individuals who had more than 90% damage to those seed regions. So we had to take these final 2 regions and look at them a little bit more exploratory in an exploratory approach. So for the regional analysis, I'll be mainly focusing on these 8 ROIs, but then towards the end of the presentation, I'll show results from these two areas, as we had to look at them in a smaller subset of participants. Okay. So our dependent variables that we were interested in, again, were the overall aphasia severity from the WOB, discourse comprehension main ideas subscore, and the discourse comprehension details subscore. And I included those 8 seed region intersubject correlation scores, as well as proportion damage to the left hemisphere and months post-stroke onset, as both of these are often associated with behavioral scores. And I wanted to, any relationship between intersubject correlation and behavioral scores, I wanted to ensure that it went beyond what is predicted from proportion damage and months post onset. And again, left hemisphere IFG and superior temporal gyrus had extensive damage to 5 participants within those seed regions. So we looked at these separately. Okay, so first for overall aphasia severity, factors that were included in the stepwise regression were overall lesion volume, once post-onset, and right hemisphere interparietal sulcus and angular gyrus intersubject correlation within those seed regions. And here are the actual versus predicted scores from this model. And the areas that were implicated are circled here along with lesion and once post-onset. For discourse comprehension main ideas, right hemisphere superior frontal gyrus and superior temporal gyrus were significant within the model, and those areas are circled here. And lesion load and months post-stroke were not included in these models as significant predictors. And then finally, for discourse comprehension details subtest, we had right hemisphere interparietal sulcus and angular gyrus as being associated with discourse details subtest. For the left hemisphere superior temporal gyrus and inferior frontal gyrus, again, we had to look at these individually. So I created stepwise regression models, including superior temporal gyrus intersubject correlation for those 11 participants that had at least 10% of the seed regions intact. Proportion damage to the left hemisphere and months post-stroke were also included in each model. And from that, we had two models that were not significant, but approaching to some degree significance, and with only 11 participants. I think this is at least somewhat promising, in that the left superior temporal gyrus was associated with behavior, particularly in overall aphasia severity and discourse comprehension main ideas subtest. Okay, for the left inferior frontal gyrus, we didn't have any significance between that area's synchrony and behavioral scores, but lesion load was significant for both of these, within both of these models. Okay, so to summarize, I know I've thrown a lot of results at all of us today. So the first aim was to investigate how a lesion can impact some non-lesioned regions, particularly within how an individual is able to synchronize compared to a neurotypical individual. So we did find a group difference between intersubject correlations, particularly in the left hemisphere, superior temporal gyrus, which is not surprising given the fact that these individuals had a left hemisphere stroke. But we also noticed or found significant differences between groups in intact regions in the right hemisphere, of course, and in bilateral visual cortices, as this was a visual task, an audiovisual task. And you can see those areas there. We had an overall difference between how well an individual, each group of people with aphasia, and how they relate or synchronize to the controls, and compared to how the controls synchronize to the other controls, in that the overall neurotypical group has greater synchrony compared to the aphasic group. And from those same scans, we also investigated global and regional intersubject correlations with behavior. We didn't find a global relationship to overall aphasia severity or any measure of comprehension, but rather this was a region-dependent relationship with behavioral scores, in that some regions that were implicated in those models are circled below, which were in the right hemisphere angular gyrus, interparietal sulcus, superior temporal gyrus, inferior frontal gyrus, and the same interparietal and angular gyrus within the right hemisphere for details. For the two areas within the left hemisphere that we had to look at a little bit more creatively, again, this was only within 11 individuals with aphasia, but we did see that there was a trending relationship between left hemisphere superior temporal gyrus, intersubject correlations with the neurotypical group, so how similarly each individual with aphasia performs to the control group, and its association with behavior, particularly with overall aphasia severity and discourse main ideas. And left hemisphere inferior frontal gyrus was not a significant predictor that was entered into any model for prediction of behavior. So I want to situate us a little bit with prior studies that have utilized this approach and where we kind of stand going forward. So earlier studies that utilize this approach in MRI scans, for example, suggest that this is a measure that can indicate how similarly individuals perceive a stimulus. So they primarily utilized online behavioral scores, so comprehension tests of the movie that they watched, for example, likability scores of the movie they watched, or even their emotional reactions or how emotional they related to the video itself. And they only looked at this synchrony score within neurotypical individuals who presumably have intact comprehension ability. So they all performed within normal limits on perceptual tasks, presumably. And this is something that is really tricky when reading the literature, as we really weren't clear on if this is a mechanism of comprehension or truly just something that is innate in an individual attending to the same stimulus. There have been a few groups that have used this approach in clinical populations, primarily in individuals with autism, but also in folks who have trait-like behavior like depression, schizophrenia, and paranoia, as well as other trait-like behaviors like cognitive approach, whether they think more hierarchically or detail-oriented, for example. But these are all traits and diagnoses that affect global processes. And the idea behind this project was to look at a group of individuals who have a very specific language deficit in comprehension ability to investigate if this is a mechanism of comprehension. So so from the prior studies that have utilized this, there have been propositions that this is a comprehension mechanism. But utilizing our sample with individuals with language deficits really helps us to look at this further. And we did find some promising results to suggest that there is some relationship between intersubject correlations and offline comprehension ability in groups with comprehension deficits caused by a lesion. And finally, just to kind of frame this up, the results from the study do suggest that there is, of course, a difference between individuals with aphasia and neurotypical controls, even in regions that are left spared from the lesion, and that this isn't a global relationship with behavior, but rather individual regions' ability to synchronize or perform similarly to the control group have a relationship with overall discourse measures and even aphasia severity. And I think that this might give us some framework in thinking that we could provide a way for us to identify certain neural targets that could be targeted for treatment, or even targets for stimulation, where we could provide external stimulation to kind of introducing how a region should synchronize to a specific stimulus. And just some limitations to keep in mind. So no study has investigated this, of course, with individuals who have a lesion, let alone different lesion profiles, which of course always throws some fun into the mix. But it would be really informative for us to have some participants with more varying degrees of comprehension deficits. So I plan to recruit some more participants who show a greater degree of comprehension deficits, so that we can get a better idea of if this truly is a function of comprehension at large. But also I want to get an idea of if we can assign certain regions to their own specific function and how they contribute to comprehension ability in comparing synchrony to certain seed regions to phoneme recognition region or scores, or syntax measures, or other like executive function measures like working memory and attention, and see if we can maybe ascribe or identify whether synchrony within certain seed regions is attributable to some of these more lower level, but also contribute to higher level comprehension ability. And then future directions, I also plan to utilize some functional region of interest in localizing certain patient-specific regions and identifying synchrony within those regions as well. To close, I want to thank everybody that has contributed to this body of work, including Stephen Wilson and Riel Stark, but also the many people within our group at USC who have been very helpful in this project, and our participants for taking the time to complete the study. Thank you very much.
Okay, thank you, Dr. Johnson. Thank you, Lisa, for your talk. That was great. We have a few questions already in the chat box, so audience, feel free to add to those. I'm going to read out the questions to you, Lisa, okay?
Okay.
So Heather Dial asks, can or has the ISC already been used to look at inter-subject correlations of within subject between regions connectivity? For example, looking within an individual at the pairwise correlation between right STG and right angular gyrus, and then at the inter-subject correlation of this correlation. I'm asking about this because I'm thinking about a way to extend beyond activity in single regions to something more network-based.
Yes, the short answer, Heather, is that's a great question. Number one, there are different measures of intersubject correlations out there, like the ones that you're describing that are more network focused, in that in an individual within area, like within STG, you can look at how that area is related to within the network, within one individual, and the network of the control group who you're comparing to. And I'm happy to send you some resources on that. I believe there's another free software or toolbox that's available for running that kind of analysis. But it's not as used within this body of work. So I've only been able to find maybe a handful of studies that have utilized this approach.
Thank you very much. A question from Kathy Torrington-Eaton. Thank you for a great talk. Did you take any behavioral measures of attention? It was interesting to see the involvement or lack of involvement of right parietal areas. Can these findings inform future directions? Yeah,
that's a great question. We did have a measure from the WACE matrices subtest, and that wasn't associated with any comprehension or any synchrony measures, which I was also very surprised about. But I really do think that, of course, it's always after you finish collecting data that you think about all of the things that you wish that you had collected. And I do think that other measures of attention or executive function would have been really interesting in looking at some of those more frontal regions or parietal regions too, yeah.
Thank you. I had a question as well. How much would a delay in the bulb response and processing affect the ISC values for the aphasic participants? So what if they have similar patterns, but just delayed relative to non-aphasic controls?
Yes, this is a great question. And something that is usually framed as a pro when it comes to utilizing this analysis technique is that they suggest that by looking within the same time-locked stimulus, you're not really seeing as much of an impact of bold within neurotypical individuals, because they're suggesting that this makes up for any issue of bold delay. But within individuals who've had a stroke, of course, I think this could be a huge factor. But when looking at this, I did run an analysis where they actually jitter the time series for each individual with aphasia to see what lines up the best. And if it's delayed, we would delay it a little bit so that it does look at the most optimal correlation, whatever jitter you assign it to. But yeah, there are ways to kind of account for that. But that's a really, really great question.
Well, thank you so much.
Oh, you're welcome.
I have another question. Would it be of interest to also compare ISCs in response to stimuli or actions from domains outside of language in these same aphasic stroke survivors to address to what extent this is specific to their impaired function or simply due to stroke in general? Right, so whether the disrupted neural synchrony is just a, it's just what happens to the brain after stroke, but does it really have to do with their impaired function?
Yeah. I kind of struggled with this towards the beginning, about just kind of conceptualizing the study. But I mean, I guess the proposition in the literature is that this is for comprehension. So the idea was to either support or refute that claim. But I do think that it will be important in future steps to see if this is just, or even looking at in contrast, if this is truly a comprehension or response to comprehension, maybe comparing it to speech production measures, for example, to show that this is not associated with one of those more motor tasks, for example.
Yep. Thank you.
Yep.
I have no other questions in the chat box for you at this moment. So unless someone starts typing right now, I think we can thank you, Lisa, for your talk.
Yeah.
And I want to remind the audience of the talk in 2 weeks by Jenny Mack. And then in 4 weeks' time, we have Professor Stefano Cappa, who will join us. And after that, we have a whole season of superb talks for you. So please check out our websites. Thank you, Dr. Johnson, for your talk today. And thanks to the audience. And we hope to see you in 2 weeks' time.
Thank you for having me. Thanks.